So, imagine a doctor sitting across a desk from someone in their 50s,
Right.
and the patient came in because they've been, short of breath for a few months. Maybe they had this persistent dry cough that just wouldn't go away.
Yeah, a really common scenario in respiratory clinics.
Exactly. So the doctor gets the scans back, and the diagnosis is idiopathic pulmonary fibrosis.
Which is, well, it's brutal. Idiopathic meaning nobody actually knows what causes it, right?
Right.
And then the pulmonary fibrosis part means the delicate air sacs in the lungs are like slowly and irreversibly turning into thick scar tissue.
Yeah.
Every single breath becomes a little shallower than the last one.
It's just a devastating moment for you as a patient because
Yeah.
there's no cure.
None.
The median survival after you get that diagnosis is 2 to 3 years.
Yeah.
And this exact conversation has been happening basically unchanged for decades.
Yeah, the drugs we have can, slow the disease down a bit,
Yeah.
but they absolutely cannot stop it.
Right. For 50 years, the underlying biology of this disease was just this impenetrable fortress. It resisted literally every approach we threw at it.
Because we simply couldn't find the right biological mechanism to target, let alone engineer the right molecule to hit that target with. The traditional tools were just failing us at step one.
But then, in March of 2026, something happened that fundamentally changed the shape of that entire conversation.
It really did.
A small molecule called NS018055 produced positive phase 2 results in human patients with that exact disease. The clinical trial data was peer-reviewed, published in Nature Biotechnology.
And what makes this unprecedented isn't just that it showed efficacy, it's the origin story of the molecule itself.
Yeah.
This drug was discovered by an AI, designed by an AI, and it targets a specific protein that nobody had successfully drugged before.
It's the first time in history a drug discovered and designed end-to-end by artificial intelligence has actually shown clinical evidence of working in human patients.
It's massive.
It is. Welcome back to Frontier Notes for this deep dive. Today, our mission is exploring how AI quietly, and then, very suddenly redesigned biology.
You have a really fascinating stack of sources for you to unpack today.
Yeah, we're pulling from groundbreaking papers from Nature and Nature Biotechnology, the 2024 Nobel Prize press releases, technical white papers from isomorphic labs, and of course the overarching script from the Frontier Notes team.
So, settle in, because we're going to look at what this all actually means for the future of medicine.
And for the next person sitting across that doctor's desk.
Exactly. But to truly understand why this matters to you and to medicine as a whole, we kind of need to bypass the basic biology for a second.
Right. We need to look directly at the computational nightmare that haunted researchers for, half a century?
Yeah, the protein folding problem.
So let's set the stage here. Every cell in your body is essentially this microscopic workshop, right?
Yeah.
Packed full of tiny, complex machines called proteins.
They do literally everything. They contract your muscles, carry oxygen, fight infections.
They even read your DNA.
Mm-hmm.
But their function is entirely dictated by their three-dimensional shape.
Right.
And I think we need to ditch the old lock and key metaphor here.
For sure. It's way too simplistic.
Yeah, because if we're targeting a protein, we aren't trying to cut a flat brass key. We're trying to design a custom, highly flexible spacesuit for a microscopic alien with thousands of moving tentacles.
I love that analogy.
Right. If you don't know exactly how those tentacles fold and shift and interact in 3D space, any suit you design is going to fail completely.
That captures the complexity perfectly, because proteins start as these linear sequences of amino acids.
Right.
But in fractions of a second, they spontaneously crumple and fold into these highly elaborate, dynamic origami structures.
Just instantly.
Basically, yeah. And predicting exactly how that linear string would fold into 3D space was considered practically impossible. The number of possible configurations for a typical protein is greater than the number of atoms in the observable universe.
Which is just an insane statistic.
It is. So for decades, researchers ran this biennial global competition called CASP, the Critical Assessment of Structure Prediction.
Right, where teams submitted their best computer models to try and predict structures that had just been mapped in the lab.
Exactly. And for years, the very best methods in the entire world were totally paralyzed, hovering around 60% accuracy.
So the structural map of biology was essentially incomplete.
Completely incomplete.
Until 2018, when DeepMind entered that CASP competition with their AlphaFold system and just shattered the existing benchmarks.
And they followed that up in 2020 with AlphaFold 2, which achieved experimental accuracy.
Wow.
Yeah, it was producing predictions that were as accurate as the painstakingly slow laboratory measurements, things like X-ray crystallography or cryo-electron microscopy.
So the 50-year-old protein folding problem was, for all practical purposes, just solved.
Solved, yeah.
Here's where it gets really interesting. Let's look at the sheer, mind-bending scale of what AlphaFold unleashed.
The numbers are staggering.
They really are. So over the previous 65 years, human scientists had experimentally determined the structures of about 200,000 proteins.
Right.
65 years. 200,000 structures. Then DeepMind just flipped a switch. They used AlphaFold 2 to predict the structure of essentially every known protein to science.
All of them.
200 million of them. They released it in a single database update completely for free. AlphaFold added 1,000 times more structural knowledge in one single moment than humanity had gathered in its entire history.
It was an absolute earthquake for the pharmaceutical industry.
I bet.
I mean, the graduate students who used to spend 4 years of their PhD just trying to crystallize one single protein to map its shape, they didn't have to do that anymore.
And just type it in.
You simply type the amino acid sequence into a web form, and the 3D structure just materializes on your screen in seconds.
Unbelievable. And that breakthrough earned Demis Hassabis and John Jumper half of the 2024 Nobel Prize in Chemistry.
Well deserved.
Oh, absolutely. But knowing the shape of a protein only tells you what already exists, right?
Yeah.
It's just reading the language of biology.
Exactly. And reading isn't enough to cure a disease. You need to write.
Right. So if AlphaFold taught us how to read the language of proteins, the other half of that 2024 Nobel Prize was awarded for teaching us how to write it.
Yeah, David Baker at the University of Washington took home the other half of the Nobel for his work in de novo protein design.
De novo meaning from scratch.
Right. So instead of looking at an existing protein and asking what is its shape, his lab asked the total inverse.
Okay.
They asked, if I need a specific shape to perform a specific function, like, say, binding tightly to a cancer cell, what sequence of amino acids do I need to invent to create that shape?
They introduced some truly wild tools to actually solve this.
They did.
One is protein MPNN, right? Which takes a desired 3D backbone and kind of works backward to give you the exact sequence of amino acids you need to make that shape physically stable.
Yes, but the tool that absolutely blows my mind is RF diffusion.
Okay, so wait, I want to make sure I have this mechanism right, because it sounds crazy.
Go for it.
RF diffusion uses the same kind of generative diffusion architecture that creates those AI images on the internet, right? Like astronauts riding horses.
The exact same underlying architecture.
But instead of refining static noise into pixels, it's refining atomic noise into functional physical protein coordinates.
Yes. The architecture is incredibly similar, but the application is, well, infinitely more complex.
Right.
In an image diffusion model, the AI learns to denoise a static image step by step. But RF diffusion was trained to denoise the 3D coordinates and spatial orientations of amino acid residues.
That is wild.
You literally start with a cloud of random atoms, just structural noise in a digital vacuum.
Wow.
And the model iteratively refines them, but it has to obey the laws of physics, accounting for bond angles and atomic forces, until that noise resolves into a completely novel, stable protein structure. One that nature just never bothered to invent.
So it's basically simulating billions of years of evolution in a matter of minutes on a GPU.
Pretty much. We can design custom binders, new enzymes, precise vaccine scaffolds.
But Baker's work on creating new proteins revealed a blind spot, didn't it?
It did. Because proteins don't operate in a vacuum.
Right.
If we want to actually cure a disease, we need to know how these proteins interact with other molecules, like DNA, RNA, or existing chemical drugs. The AI needed to understand the entire biological ecosystem.
And that's exactly what AlphaFold 3 was designed to do when it dropped in May 2024.
Yeah, it was a joint effort between DeepMind and Isomorphic Labs. They moved beyond just isolated proteins to predict the joint structure of biological complexes.
Meaning it could predict how a protein folds around a small molecule drug.
Exactly. And less than 2 years later, in February 2026, Isomorphic Labs revealed IsoDD, their drug design engine.
Right.
And it literally doubled the accuracy of AlphaFold 3 on completely novel targets.
So I saw the term cryptic pockets in the isomorphic white paper, and I really want to make sure I'm understanding the mechanism here.
Oh, cryptic pockets are fascinating.
It sounds like a secret trap door. I'm assuming this means the AI is mapping physical vulnerabilities on a protein that are completely hidden when the protein is just floating around on its own, but then they suddenly open up when a specific molecule forces a structural shift.
You've captured the exact dynamic.
Oh, nice.
Yeah, traditional methods look at the static empty protein, right? So they miss these pockets entirely because they physically aren't there yet.
They don't exist until something bumps into them.
Right. But ISO-DDE learned the dynamic physics of the protein well enough to predict exactly where these hidden doorways would form under the right conditions.
Wow.
They found a novel cryptic site on a protein called cereblon, which is actually part of the body's natural waste disposal system.
Okay, so if you can drug that pocket.
You can basically trick the body into destroying disease-causing proteins.
Mm.
Investors saw this capability and backed Isomorphic with $600 million.
600 million, that's huge. But to see the real-world human stakes of all this AI acceleration, we have to return to our cold open.
We do.
We have to go back to that patient with idiopathic pulmonary fibrosis.
Because to understand the magnitude of what happened with that patient, we had to look at how totally broken the traditional drug discovery pipeline actually is.
Oh, it's a nightmare. The standard process is, first, you spend years trying to identify a target, usually a protein driving the disease.
Right.
Then, you spend years screening millions of existing chemicals, just hoping to find one that binds to it. Then safety testing. Then human efficacy trials.
And the entire process typically takes 10 to 15 years.
10 to 15 years. And it costs well over $1 billion. And even with all that, fewer than 1 in 10 drugs that enter clinical trials ever actually make it to the market.
And for idiopathic pulmonary fibrosis, the failure was happening right at step 1.
Right.
Researchers literally couldn't identify the correct biological mechanism to target.
Enter in silico medicine. They're a biotech company based in Hong Kong, founded by Alex Zavaronkov. And they decided to aim 2 distinct AI systems directly at this untreatable disease. The first system is called Pandomics. It's a target identification engine.
But it didn't just look at the lungs.
Right. It mapped out the entire cascade of biological signals across different tissues that tell scar tissue to grow in the first place.
It digested massive biological databases, genetic profiles, millions of text publications. It basically mapped the graph network of the whole disease.
Just looking for the central junction box.
Exactly, the primary node where all these different disease pathways intersected. And in 2019, Pandomics flagged a target that humans had completely overlooked.
A protein kinase called TNIK.
Yes, they found their hidden target. Next, they fired up their second AI system, Chemistry 42.
And this is the generative AI for small molecules.
Right. So instead of sifting through libraries of old drugs hoping for a lucky match, it generated a brand new molecule from scratch.
Wow.
Optimizing it for safety, metabolic stability, and its ability to perfectly bind to that TNIK junction box.
The timeline here is what should really keep pharmaceutical executives awake at night. Because from the start of the project to nominating a final, highly optimized preclinical drug candidate, it took them a staggering 18 months.
18 months to accomplish what traditionally takes 4 to 6 years of trial and error.
Just incredible. They named the candidate INS-018055. It cleared phase 1 safety trials and moved straight into phase 2A trials in human patients with IPF.
And then in March 2026, the results hit Nature Biotechnology.
Right. So the primary goal of phase 2A is proving the drug is safe and well tolerated in the target population.
Which it cleared. But the secondary endpoint is what actually made the headlines. It showed clinical evidence of efficacy.
And the specific numbers here are just incredible. So they measured forced vital capacity, or FVC.
Right, which is a measure of how much air you can forcefully exhale.
Yeah, it's the ultimate metric of lung function for someone whose lungs are turning to scar tissue. Over the 12-week study, the patients receiving a placebo saw their FVC decrease by 62.3 milliliters.
So their decline was continuing exactly as the grim prognosis predicted.
Right. But the patients taking a 60 milligram dose of the AI-designed drug, INS-018055, they didn't just plateau.
No.
They saw an improvement in FVC of 98.4 milliliters.
Their lung function actually improved. I mean, a biological process that we believed was a one-way street was actively reversed over a 12-week period.
Okay, let's unpack this because this is huge. They mapped the pathways with AI, found the hidden target with AI, designed the molecule from scratch with AI, and it reversed lung decline in human patients.
Yeah.
It's really hard not to look at this pipeline and think AI just fundamentally solved drug discovery.
Well, this raises an important question, right? I wouldn't go quite that far yet.
Really? You don't think this is a complete threshold crossed?
It's a monumental milestone, absolutely. It is the first definitive proof that an end-to-end AI pipeline can produce a biologically active, clinically beneficial medicine.
Okay.
But we have to look at the reality of a phase 2A trial. It's 71 patients over 12 weeks. Biology is infinitely messy, and IPF is incredibly complex. It's entirely possible this was just an incredibly accurate dart throw guided by a brilliant algorithm.
Just a lucky hit.
It could be. The drug still has to survive much larger Phase 2b and Phase 3 trials to prove long-term efficacy and safety across a very diverse population.
That's a vital pushback, actually, because while AI has completely warped the timeline of the early computational stages of drug discovery, it hasn't changed the fundamental biological speed limit of the human body.
Exactly. The contract and timelines is staggering now.
Yeah.
Target identification. AI compressed that from years down to months.
Designing the candidate molecule was compressed from years down to weeks.
Predicting the 3D structures and interactions, compressed from months down to seconds.
But the clinical trials, Phase 1, Phase 2, and Phase 3 combined, still take roughly a decade.
Right. Even with in silico's hyper-fast 18-month sprint to engineer the molecule, it still took them a total of 7 years to reach that Phase 2 readout. Because the molecule still has to sit inside a human body. You have to recruit the patients, administer the drug, wait to see if adverse events emerge, and track lung capacity over months and years.
Human biology just hasn't sped up just because our GPUs got faster.
AI compresses the generation process everywhere it touches, but the validation process remains resolutely human, and humans take time.
Which brings us to the grand pattern we've been tracking across this series, really.
Yes.
In our first deep dive, we saw that the central bottleneck in science moved from the production of research to the verification of it.
Right.
In our second deep dive on AI safety, we saw the bottleneck move from designing capable systems to deciding what values they should actually align with.
And now, today, we see the exact same shape emerging in medicine.
The bottleneck of drug discovery has officially moved. It is no longer generation finding the hidden targets and inventing the molecules. The bottleneck is validation proving in actual human bodies that they actually work.
It's a pretty profound realization. AI has essentially conquered the computational half of the pharmaceutical industry.
Right.
But the biological half is still this slow, physical, very human endeavor.
So let's bring it back to where we started. That patient sitting in the doctor's office looking at those lung scans.
Yeah.
The reality is INS 018055 isn't approved yet. As we sit here, the massive Phase 3 data is still ahead. The drug could still fail in a broader population.
It absolutely could.
But for the very first time in 50 years, that patient's doctor has a reason to point to a candidate molecule that came from a fundamentally new place.
It wasn't found by just randomly screening thousands of old chemicals.
No, it was engineered intentionally by an intelligence that could map the intersection of biological pathways we couldn't even see.
It really represents a totally new era of hope. We are finally writing the language of biology.
But I want to leave you with one final thought to mull over today. If AI has already conquered the generation of new drugs, moving the bottleneck entirely to slow human trials, what happens to medicine and to us when AI eventually finds a way to simulate the entire human body at a molecular level and disrupts the validation bottleneck itself?
No, that is a big question.
We'll let you think about that. Thank you for joining us on this deep dive. Next time, we'll be exploring how AI is about to change the shape of computing itself. We'll see you then.